So let's talk about peptides, an important thing to learn about if you're learning pretty much anything about biology. So what even are peptides? Well, peptides are polymers of amino acid subunits, and we call the bonds that link each subunit together the peptide bond. Chemically speaking, the peptide bond is an amide linkage, which is a connection between the carbonyl carbon of one amino acid and the amine nitrogen of another amino acid. Looking at it from a synthetic perspective, this means the carboxylic acid group of one amino acid subunit is linked to the amino group of the next amino acid subunit, and so on and so on and so forth. The bond forms when the lone pair of electrons on the amine nitrogen nucleophilically attacks the carbonyl carbon on the carboxyl end. One of the carboxylic acid's oxygen atoms leaves as a water molecule. So this reaction is technically an example of a condensation reaction and can also be classified as a dehydration reaction. This is also an example of a nucleophilic acyl substitution mechanism, which we often see in reactions involving carboxylic acid derivatives. With that said, when our cells actually make and synthesize proteins, it's not quite as simple. Ribosomes mediate the process of making proteins by involving both transfer RNA and messenger RNA. But chemically, all the ribosome does is catalyze the peptide bond formation. The basic logic of nucleophilic acyl substitution involving an attack by a lone pair of the amine nitrogen on the carbonyl carbon still applies. The details don't need to concern us here. Instead, it's just worth being broadly aware of the fact that reality is a little more complicated than this simple little mechanism would imply. The peptide bond is broken through a hydrolysis reaction, which is simply the reverse of the condensation reaction. A molecule of water attacks the carbonyl carbon and kicks off the amine group, generating two new peptides or amino acids, one with a new carboxylic acid group and one with a new free amine. This reaction is how proteins are broken down, and it is very important. So it gets its own special name, protealysis. Interestingly, hydrolysis of peptide bonds is energetically favorable, but it's really, really slow under physiological conditions, which is a good thing after all, since, we wouldn't be in good shape if the peptide bonds holding together the proteins in our body were spontaneously breaking all of the time. Living cells, however, do need a controlled way to break peptide bonds. So specific enzymes known as proteases exist to do just that. Some proteases are known as digestive enzymes because they break down the proteins we obtain through our diet. Strongly acidic conditions can also facilitate peptide bond breakdown, but even in the stomach with its pH of 2, specialized digestive enzymes like pepsin are largely responsible for this task. One reason why peptide bonds are so stable under intracellular conditions is that the peptide bond actually has partial double bond characteristics because of the delocalization of the nitrogen's lone pair of electrons. We can illustrate this by drawing a resonance structure where nitrogen's electrons are donated to form a double bond between it and the carbonyl carbon, with the carbonyl's pi electrons placed on the oxygen atom. This means that when we average the structures, the oxygen will have a partial negative charge and the nitrogen will have a partial positive charge. Conjugation requires p orbitals, which means that the molecular geometry of nearby atoms is planar, so the peptide bonds don't rotate freely, a property that contributes to the structural stability of the three-dimensional polypeptide structures. Now, our discussion of peptide bonds has been short but sweet. Peptide bonds are structurally simple, but they are a remarkable intersection point for a surprisingly diverse set of concepts from general and organic chemistry, ranging from the nucleophilic substitution mechanism used in their formation and breakdown, all the way to the phenomena of resonance stabilization and the impacts of the planar geometry of these bonds. As you study these concepts in more depth, keep peptide bonds in the back of your mind as a real-world example.